Good afternoon, everyone, and welcome back to yet another exciting session of the Clinical Assessor Shortcast. We are now on top of the hour, and we will start our lesson for today, which is lesson 11, titled Clinical Data, Safety, Risk Management, and Mitigation. A quick sound check. Can someone confirm that you can hear me? Peter, can you hear me?
Yes, I can hear you. Yes.
Thank you very much. So just a reminder that this meeting is being recorded, and the recording will be loaded on the Moodle platform for future use and referencing. Just to go over the agenda for today. Before we do that, some housekeeping rules to remember that all students must remain muted and your videos must be switched off, but you are most welcome to type in your questions or any comments related to the lesson in the chat box. You will remember the lesson that we had last week, which focused on clinical review and clinical data for efficacy and safety. And note that the lesson ties in very well with today's topics. So our first speaker for today will be Peter Stonia. He will take us through the first presentation titled Clinical Data, Safety, Risk Management, and Mitigation. So just a quick introduction for Peter. Professor Stonia is based in the School of Cancer and Pharmaceutical Sciences at King's College London. He serves as the Director of Specialist Training at the Faculty of Pharmaceutical Medicine at Royal Colleges of Physicians in the UK. And he is the National Program Director for Pharmaceutical Medicine Specialty Training. He's been with the Clinical Assessor course for, I believe, since its inception, and we are very grateful to have him take us through his very enthusiastic talk today. Without further ado, over to you, Peter.
Thank you, Unathi. I'll just share my screen.
It is coming through. If we can just put it on slideshow. Perfect. Thank you. Over to you.
Thank you. Thank you. Well, good. We got sound and everything, right? Thank you. Well, good afternoon and good day to everybody. It's very good to be here again to talk to you. And today we're going to cover this topic of clinical data, safety, risk management, and mitigation. It's one of the few opportunities in the course to bring together a discussion on safety, which is such a key part of the regulatory assessment for licensing, as the opposite side of the coin to efficacy. So there's quite a lot of information to impart here. Some of it we will speak to you about. Some of it is on supplementary slides and presentations for you to follow up on with some self-learning this week. But we hope overall to get through the large amount of information that we would hope to present to you to make this week interesting and worthwhile for your course. So the aim of the presentation is made in two parts. Firstly, by myself. Secondly, by Dr. Assem El-Baghdadi, our colleague. And the aim of this week really is to understand how clinical data informs the ongoing risk management and mitigation of risks of products in development and post-authorization, to consider the safety and risk management approaches used in medicines development and post-approval, to place benefit risk assessment as the basis of regulatory decision-making and recommendations, and to address matters of risk management and mitigation. But I'd also like you to consider the following points as well, that safety and risk assessment is a continuous process throughout the product life cycle. It's not like many of our other topics that we've discussed in this course, where you have a stop at the point of submission for regulatory assessment. Safety continues throughout the whole life cycle, including through the regulatory assessment process and into the post-marketing phase. So I think we've got to accept that this is a continuous process. It's rather different from some of the other topics that we've had. The second point is that regulators and sponsors interact on safety on an ongoing basis, both pre and post authorization. This is safety is a topic where there is regular and ongoing interaction between regulators and sponsors. So again, that is an important point to consider. The third point to consider in this is that the whole history of safety and pharmacovigilance and the coming of risk considerations, risk management, has moved the whole topic on from over many, many years, from a topic of crisis management, safety disasters, and regulatory reactions to those disasters. From crisis management to risk management. So we're now very much more concerned with prevention and with undertaking a risk proportionate approach to safety assessment. The other point to make here is that benefit risk assessment by many stakeholders is a continuous process also. We've had from 2 weeks ago the presentation of the universal methodology for benefit risk assessment, which is an acceptable framework and tool for benefit risk assessment. But that is only one part of the whole process of benefit risk assessment, which goes on all the time by many stakeholders without necessarily the use of tools throughout. But it is an important assessment to understand and for people to be able to make, because the final point is that a positive benefit risk balance, which is a judgment call, it cannot be calculated through computers, is the basis of product licensing and the maintenance of products on the market. So the benefit risk assessment and benefit risk balance are very important outcomes of the pharmacovigilance process that we'll be discussing. So the topics to be covered today cover an introduction. We're looking mainly at medicines development and what contributes to the safety assessment of a product. And we go on to talk a little bit about how pharmacovigilance process and methodology comes into this, but that's not the main point of this presentation and this afternoon. It's really to look at the clinical data that emerges from medicines development, both pre and post marketing, which contributes to the safety assessment of a product. There are some issues this afternoon which go into more detail about some of the activities that can take place for gathering safety data. And but we really have to put these. We don't have the time to go into the detail of these this afternoon in the presentations, but they're very important topics to know about in terms of what safety data emerges from them, which then contributes to the overall benefit risk assessment for a product. And these are put here in italics for now. Testing for specific safety issues, the question of data monitoring committees, what safety sections of clinical trial documents produce for us, and the very important topics of the development safety update reports and periodic safety update reports and PBRs, which are all specific topics for discussion, but they here are put aside as a separate set of PowerPoint slides for you to work through yourselves outside of these presentations, but they are part of the information which we want to impart for you this afternoon. Finally, risk management and mitigation will be a large part of the presentation by Dr. Baghdadi later. Just to set us off, let's look at the information that is required for government approval for prescription use in humans of medicines, which is not different, whatever the therapies are being developed. Firstly, manufacturing quality. Can the product be manufactured to a consistent quality and required quantities for patients? Secondly, immediately, safety and tolerability. Is the product safe and well tolerated in the patients to be treated? Thirdly, efficacy. Does the product, when administered to patients, produce the beneficial therapeutic effects required? And finally, back to safety again, in part, benefits and risks. Does the product bring significant clinical benefits to the patient whilst minimizing the risks of treatment? So these are essential information which is required for approval of prescription medicines for humans. So why is safety assessment important from a medical point of view? Well, here we show that since 1999, at least 17 drug products have been withdrawn from the UK market alone, all but one of them for safety reasons. So why is safety important from a practical point of view? Safety assessment is based on the detection, assessment, and prevention of adverse effects or any other drug-related problems. That's a kind of definition of pharmacovigilance, really, but it's actually an important thing to say here. Data that emerges for this could be direct or indirect toxicity. It could be signs and symptoms of an adverse effect. It could be an indicator of a potential adverse effect which is going to arise. It could be reports of adverse events and serious adverse events themselves. And later on in the product life cycle, there's the suspected adverse drug reaction reports to consider too. This data that we get is subjected to processes of pharmacovigilance, the creation of case studies, individual case safety reports, the aggregated data from those reports on databases, the undertaking signal detection and evaluation, and then that leads into immediately into the benefit risk assessment process, and ultimately then important communications on safety and risk to healthcare professionals, patients, and to the public. It is really a description of the process of pharmacovigilance, which is applied to all data which emerges from drug development stages or from any other source of potential safety data. And data collection like this throughout the life cycle of the medicine is analysed regularly to identify and characterise the risks for a product. So there's no question of it stopping at any point. But nowadays it's possible to identify certain leading indicators of safety problems, and one of these is known as a sentinel event, which is a serious adverse event that is not necessarily drug related, adverse events are not necessarily drug-related, but has been associated historically with adverse events, adverse reactions to other drugs, and is therefore worthy of heightened pharmacovigilance. And some of these sentinel events you will recognise here as being very serious conditions which occur and are associated with and produce adverse reactions. So it's important to always, there is a need for heightened pharmacovigilance for these events. So the first question we want to ask today is, in medicines development and post-approval, what contributes to the safety assessment of a product? And I'm just going to go through now where in drug development, in the launch and the marketing of a product, is data generated in terms of its safety assessment. And we can see here a stylistic diagram of the drug development stages from discovery and preclinical development through clinical development into registration and marketing. You will be extremely familiar with this diagram, I'm sure. But I put it here to indicate that throughout the drug development process, we have to look for data which indicates a safety issue. It's not just a question of looking in the clinical phase or in the clinical trial phase or waiting for post-registration to look for this data. It goes on throughout the whole development stage in terms of the production of data, which then can lead to information on safety, and that would lead into the assessment of safety data and lead into the benefit risk assessment for the product. So this is a representation of that, but I'm going to look at it in more detail from a cartoon point of view, a diagrammatic point of view, to show, to just indicate very briefly, because we don't have a lot of time to cover the detail of all of this, but where in drug development, in the launch and marketing of a product, the data generated in terms of its safety assessment. So we can look first at the early stages, the preclinical stages, the non-clinical development and the preclinical development, which are different. The non-clinical development stage is the very early stage of laboratory work, of scientific work in research to look for hits into disease targets, for looking for hit generation that we can then further develop into lead molecules for the possibility of product development. And this goes on in the non-clinical and the preclinical stages before the medicine is introduced to humans. And the data and information from non-clinical and preclinical stages is listed here, and I'm not going to go through it all for every slide because there's a lot of it. But I want to point out that for any data that is produced from any of these development stages, all of it could be potentially safety data, which needs to be further looked at and further investigated. So we have, for example, the preliminary pharmacokinetic data and single dose pharmacokinetics in animals. The animal work occurs largely in the preclinical development phase, when we're looking for concentrating the hits and more selectively developing lead compounds for potential clinical development. This is where the animal work takes place. So we have single-dose toxicology as well, and biodistribution. We have immunogenicity potential is looked at this stage. And we then have review of proof of concept and in vitro and in vivo data and adverse effects observed from the animal work. And we have the design of the target product profile, which is best done at this early stage. And we also have the production of the draft summary of product characteristics, which will detail some of the potential safety issues. The next stage really is coming up to filing for undertaking clinical studies through an IND or through a clinical trial application. And on the left here, we can see some of the data which has been accumulated at this stage of the CTA filing in terms of toxicology studies and dose kinetics in animals and putative dose ranging for humans, and sort of also regulatory matters in terms of pre-submission meetings, orphan drug considerations, and preparation for filing. And then for the actual submissions itself, which you see on this extra box on the right, we have the data that's been accumulated so far, which can be submitted for the application for a clinical trial clinical trial permission or an indication for a new drug submission. So preclinical data, the investigator brochure, clinical trial protocol, the risk management plan, and information on formulation and manufacturing. All of this is the accumulation of data to date, to this part of the process. And then we can start in clinical studies, and again, these are familiar to you, I'm sure, going through the phases of clinical work. Phase 1, usually in healthy volunteers, except in oncology, obviously, when patients are used. So we have single dose studies, repeat dose escalation, and so forth. And we also have the development of the development safety update reports, which coordinate all the safety data on an annual basis. And we have teratology studies and further toxicology studies and so forth. So all of this can produce potential safety data. And the same in phase 2. As we go through this phase now, we're moving into the clinical studies in patients with the target condition, phase 2A studies, clinical studies. We're looking at dose ranging, we're looking at dose finding and dose ranging. We're looking at more specific safety assessments with the emergence of adverse event reports, and so forth. And we're looking here at the proof of concept of the drug as well. And obviously a safety assessment at this point is extremely important as to whether this represents a go, no-go decision for further development. And then if the drug is okay to go forward, it would go into phase 3, which are the confirmatory studies, really, as they're called. So we're looking at the phase 3 studies, safety assessments, further toxicology, which is taking place later in the product life cycle. And we're looking at regulatory issues such as the development safety update reports, which are a moving, developing set of reports to indicate how the safety data has accumulated and developed. We have pre-submission meetings with the regulators, between the regulators and the sponsors. We have a risk-benefit assessment, and we have the generation of the summary of product characteristics and package inserts initially through the product information. So this is a very important phase, obviously, of development, because this is pre-filing for the marketing authorization application or the new drug application, the NDA. So that's a special situation which will be described later as to what data goes into the marketing authorization. But clearly, it's an accumulation of all the safety data that's been derived from the development program. And of course, it's not complete because the development program itself doesn't produce a complete set of data to be able to, say that the product is safe. But in post-approval, the process goes on to capture safety data from phase 4 studies, from pharmacovigilance itself, and that is all summarized in safety update reports and periodic safety update reports and what are now called periodic benefit risk evaluation reports, PBREs, to bring in the concept of benefit as well as risk. And then we have the risk management plan. Risk mitigation activities are beginning to be described. And then we have market support with advertising and promotion, which goes on in parallel to the actual scientific and medical development of the products. And we have publications and we have new indications. We have a lot of activity going on in the post-marketing phase, particularly the early post-marketing phase, which affects the safety of the product. So in medicines development and post-approval, what contributes to the safety of a product? Well, in summary, the drug safety data and information are contributory during all the phases of product development and marketing. It's not just about collecting safety data and reviewing it, but it's also about considering all aspects of the process to the market and beyond from a safety perspective. And this gives rise to the concept of a continuous benefit risk assessment by regulators and by practitioners and by anybody who is involved in the medicines business, really, because who have an understanding of the benefits and risks which have to be evaluated and assessed. So I'm just summarizing here those other activities which we're not going to make a presentation of, but which I refer you to in the supplementary slides which are on the Moodle platform. And these are further activities in the development stage which could give rise to safety data, such as testing for specific safety issues. Examples here are looking for the incidents and the issues to do with drug-induced liver injury, which itself is such an important or has been such an important reason for drug withdrawals over the last decade, really. So looking for indicators of drug-induced liver injury is really very important for a product. Specific product. And the same thing for if we detect QT interval prolongation and proarrhythmic potential in a compound, then this can be studied now quite specifically during the development phase to see what the implications of this might be for the product. Secondly, the data monitoring committee is an important regulated activity, introducing a group of experts into an independent data monitoring committee in clinical trials. Not all clinical trials, but the more important pivotal clinical studies have data monitoring committees allied to them who make decisions on the emerging safety data and whether and how that impacts the future of the clinical trial, whether it's continued, whether it's halted, or whether it's terminated. Halted temporarily, for example, or whether it's terminated. So there's a lot of important points to make about data monitoring committees, which you can learn about by looking at the slides on the Moodle platform. Clinical sections of clinical trial documents that inform the benefit risk balance are also extremely important, and particularly the protocol and its amendments And secondly, the investigator's brochure, which is a very important document in the pre-licensing phase for accumulating all the evidence from the preclinical work and all the safety data that emerges into an investigator's brochure. And then we have, as we've said already, the drug safety update reports and the PSURs and PBRs. The drug safety update report is actually called, there's several names for this, but it's basically developmental safety update report. Actually, it's not drug safety update report. And see full descriptions of these very important topics in the supplementary slides. Finally, on this slide for additional data is the benefit risk assessment process itself should be considered here. So to conclude this presentation, we just say that all safety data that occurs enters a pharmacovigilance system and undergoes analysis, assessment, and evaluation. And a pharmacovigilance system, whether it's in a sponsor organization or actually in a regulatory body, it has different aims, but it is actually concerned with the same activities. Number one is case management and the creation of individual case safety reports, what I would call the units of value in early pharmacovigilance. If the quality of case safety reports is poor, then it's likely that this has an impact on the quality of further pharmacovigilance down the line. And, this is an important thing to keep in mind. So you must have, for a good case, you must have adequate data on the event, the clinical detail of the patient, the product information. You must then create from as full information as you can possibly get the case narrative and the pharmacovigilance commentary, aiming always for a causality assessment of the case. And then the next stage of PV is case aggregation, databasing of cases, so that then you can work up activities such as signal detection and workup and evaluation from the aggregated cases. So this is where the pharmacovigilance system builds up then to produce data which can go into a benefit risk assessment. And ultimately then when that's agreed, when that's proven to be positive and there's a drug available, then all of this safety data can be communicated through many channels for communication of safety data to the healthcare professionals, patients, and the public. But case reports are often incomplete, as I've said, and this is really a big difficulty with pharmacovigilance, is actually making sure that there's complete and adequate data on the case. It's important to look at relatedness and unrelatedness in terms of causality for the product, because only adverse events that are at least possibly related to drug administration should be considered as adverse reactions, for example. But moving on from there, we then enter an activity which always takes place at this point, which is causality assessment, as the method by which the extent of the relationship between exposure to a drug and a suspected adverse reaction is established. There are many factors to consider in causality, and these factors can be put into algorithms such as the Bradford-Hill criteria, the Naranjo algorithm, the WHO algorithms, to give some sort of measure of the intensity of the event to have a causal relationship with the actual drug administration. So that's a very important part of pharmacovigilance at this point. But it also involves quality management, and the quality of causality assessment depends on the quality and completeness of the case report and the follow-up reports, the effectiveness of the reporting system, the quality of the causality review process, and poor quality causality assessment can lead to erroneous conclusions of safety. So there's a big caution here that although the ICSR is a unit of value in pharmacovigilance, so therefore poor quality ICSRs impact the quality of subsequent PV activities. And that's where I would like to conclude my part of the presentation, to say that we've now accumulated a lot of safety data, and we've put it through a pharmacovigilance system, and we've come out with findings which can affect the benefit risk assessment for the product, and ultimately it's a licensability really, and its continuing maintenance on the market when it gets there. So there's additional considerations for vaccines, but I would refer you to this, to the paper on the Moodle platform, to look at vaccines pharmacovigilance. So that is the end of my presentation. Thank you very much for listening.
Thank you very much, Peter. We have a comment on the chat for you. It says from Priscilla, greetings. Thank you, Prof. Stonia. Excellent presentation. It's my favorite subject. Thank you very much for that presentation. And if anyone has any other additional questions for Peter, you are welcome to add them on the chat. We will make sure that after the Assam's session, we tackle all the questions. And Peter, if you can also maybe just type on the chat just to respond to the questions. We are short for time, and we will now be moving on to handing over to Assam El-Baghdadi. I'm not sure if Peter, you would like to introduce him, or should I go ahead?
You go ahead. He's
not a problem. We are very happy to have Dr. Assam El-Baghdadi with us today. He will be taking us through the second part of the lesson, and I believe it's a continuation of what Peter has presented. And his title is Clinical Trial Development Safety. Dr. Assam is a qualified pharmaceutical physician with over 25 years of experience in medicines development, global research, and implementing international health and clinical strategies. That is reflected in the founding of the Alpha Beta Pharma that was founded in 2009, which is a consultancy for drug development. His interests are mainly in the Middle and Far East. We are very excited to have him today, and we are looking forward to what he is going to share with us. Over to you, Dr. Assam.
Thank you very much for this kind introduction, and thank you for the invitation.
Thank you.
Good afternoon, everyone. I didn't hear anything. Can I hear you guys?
Afternoon. We can
That's
hear
good.
you.
That's good. Yeah,
Yes.
just making sure you're awake. Okay.
Yeah,
good.
we all are here. Your
right.
slides are on.
Yeah. Okay. And I will ask you while I'm going, so please keep answering and let me know just to make sure that you are awake. Okay.
Right. Not a problem.
Excellent. So this is a continuation to Peter's presentation, and it's about the safety, risk management, and mitigation. So what is that? So the benefit risk, and it is always written like this: benefit first and then the risk. So it is not risk benefit, it is benefit risk, okay? And it is assessment. It is not a balance, it's not a ratio, okay? Sometimes you find it in manuscript or in sometimes in books. Benefit risk ratio. It is not a ratio. If it is a ratio, it has to be a number coming after that. And this is not a number, it's an assessment, okay? So I just want to make this first at the beginning. Okay. With the benefit risk assessment, There are different behaviors because we are different people. When we say different people, it means we are different agencies, we are different countries, and each country has an attitude towards the benefit risk. So you need to remember that, okay? In theory, every agency has an attitude to this benefit risk, right? Not each department, maybe you could have a little bit difference, but at the end, all of them needs to follow the same attitude or approach, okay, of the benefit risk. There are three different risk-taking types. So those who are risk avoider or risk averse, so those trying to avoid all the risk as much as they can, and those who are trying to reduce the risk, okay, and those who are trying to optimize, if you would say. What's the meaning of reduce and optimize? So optimize, this means that they accept high risk, but they are trying to make sure that it is a little bit, the risk is as less as they can, but there are still risks involved. But in the reducer, so actually the risk is not high, but it is maybe it's in the little bit. What do you mean by that? And can we jump from one section to another, from one group to another? Yes, you can. You can, of course, jump from risk avoider to risk optimization. It depends on what? It depends on the product that you are working on. It depends on the indication you're working on. It depends on the population you are assessing, okay? So you could have, or let me ask you this question. So can you have one product that it is assessed differently in different indications? The answer is come on, guys. I'm waiting for an answer.
Sorry, Assam. Our students
are we muted?
Yes,
Okay.
but we are having answers on the chat, and it seems like we are having a yes. We are getting a yes.
Yes, exactly. Yes. Well done. Okay. Thank you for assisting me. Please, if anything, I cannot see the chat, so if you can always do that, this would be great.
Not a problem.
Yes, of course, it can. How can this happen? You have a product which has the same mode of action. Okay, we agree on that. So you can assess its mode of action and its risk and so on. But it's in different indications. So one indication is acute and the other indication is chronic. Different population. So one population is young, another population is old, right? And each one of them has different category and different way of assessing and different way of accepting the risk. Okay, so you cannot accept something, for example, for psoriasis, the same risk that you accept for oncology. Of course, with oncology, again, if it is life-threatening or it is, you can accept more risk. But in something which is not life-threatening, you could accept a lesser risk. Okay, right. So for clinician in the clinic or in the hospital, they are always performing the benefit risk, always performing this assessment, sometimes subconsciously. So they are not thinking of it, but it is ongoing in their head. How do they do that? Every time they write a prescription, they think about that. They think about this patient, I saw him before, and I prescribed this 2 months ago. How is he doing now? How is he suffering? Is he suffering? And how much is this suffering? And so on. Before a clinician write a prescription, always ask the patient, how did you getting on with the treatment so far until I see? Sometimes also a clinician talks either in a journal club or in a meeting, a regular meeting in the hospital or in the conferences. How about this? Tell me your experience. I give you my experience and you tell me your experience. I say, well, when I treated this patient with drug X, suffers XYZ. But when I titrated over 3 days, actually these things didn't happen before. Ah, okay, now I learn. So always there is assessment, always there is assessment. The benefit risk assessment is not written in a stone, okay? It changes over time, and it changes from one patient to another, from one clinician to another, and so on and so forth. For a pharmaceutical physicians like myself and Peter, for example, when we sit to write a protocol, we need to consider this in our head, always considering this in our head. And in early development, we don't have information. Only the information we have is coming from the lab and coming from animal, for example, okay? And we try, and when I say animal, it has to be the proper animal, right? So it's not just animal, it has to be the proper animal that you can extrapolate to human. And this is very important. And this depends on what? It depends on the disease model, and it depends on the mode of action of that product. And you say, but I am a regulatory assessor. I need to know these things. Of course you do. Okay. And don't forget, you are a scientist. All right. You have a science degree. So in theory, you should understand these things. If you don't understand, ask. Ask your colleague. Okay. Ask someone else. Ask a pharmaceutical physician. Okay. But you need to keep these things in your head, and you need to see evidence of this stuff. All right. Okay. So we're talking about extrapolation in the early development, that you don't have much information at hand. So you have to work your way on the available lab, non-clinical data, animal, in vivo and in vitro, and the healthy volunteers if they are in. Okay. And then it's the same. When I sit down to write an informed consent form, I put everything known to date about the benefit risk, everything for the patient to understand what exactly they're getting into. And this is what meant by informed consent form, which means the patient is informed of everything before they make their consent. Okay. Sometimes, and you know that, sometimes in the middle of the study, more information came on, and the sponsor and the pharmaceutical physician have to update that ICF and go back to the regulatory authority and say, we have new information, this is the information, and this is how it is reflected in the ICF. Okay? And the same happens when we prepare for the submission at the end of the all clinical trial. So again, we put all the information that we have, all the accumulative data. So I repeat again, the in vitro, in vivo, non-clinical, clinical, with all the phases that Peter was talking about, the healthy volunteers, and then the patient, proof of concept, and phase 3, how many trials you have, all of this will be put together. These are all these are all the clinical data that you have. But there are other data that you could get to talk about, like what? Data published, publication in journals, internet, spontaneous reporting from patients already, if the stuff is already on the market. All of these things, any information that you can gather and accumulate in order to make an informed benefit risk assessment. So the benefit risk assessment is the central part of the pharmaceutical medicine, and they have to remember that everyone has different perception of benefit risk. This is absolutely crucial. So when you find a drug that has been approved by the FDA, you need to think it is the FDA. FDA does not reside in Zimbabwe or in Tanzania. Okay, they are working with the background of patients of the US. So does this mean, Asim, that my benefit risk assessment could be different than theirs? Of course, absolutely. All right, absolutely. So you need to think about that. So can I disagree with another regulatory agency assessment? Of course, this is your work. Otherwise, you don't have a job. Okay, that will already take whatever they say, and that's it. This is the gospel, but it is not. Okay. Right. These are some risks. I just put it here, and of course you can add as much as you can about the risk. So the risk is, I just want to demonstrate that the risk is not just adverse event and serious adverse events, adverse drug reaction. There are many things. So you have lack of efficacy, discontinuation, all of this interaction. And interaction, this depends on the polypharmacy, for example, and it depends on the population. So if you have young population and old population, so here you could have two different benefit risk assessment for two different indications and population and indication. Also, something like overdose and toxicity, or resistance. So again, all of this would be different from agency to another. So when we talk about ethical concerns, what is ethical in one country could be, and sometimes not ethical at all in another country. So you need to think about that. Here I'm trying to demonstrate the point that you do not accept assessment of another agency as it is done deal. Financial burden. So again, some agencies do not, or some countries do not have the financial burden that your country has and your agency. Okay, right. And then you submit for the license. When you submit for the license, this information sponsor has to submit to you. And this is what I said earlier, the non-clinical data, the clinical PK and PD. And you say, can I request PK and PD? Yes, of course. As you remember what I said, you are a scientist. You need to make up your mind about this assessment. So you can ask. So you say, but how about if the sponsor submit data that I want more data? That they haven't done this analysis. Of course, it is your right to go and ask the sponsor, I need more analysis. I need more data, okay? Of course, with the justification. So it is not you being silly, and also in the same time, the sponsor not being silly to say, I cannot do this. Why you cannot do this? So it has to be a scientific justification. It's a dialogue, guys. It's a dialogue between you and the sponsor. And the dialogue means you say something, they say something back. So it is give and take. It is not one way direct. Okay. And here are just the risk management plan. What is that? We're going to talk about this in a minute. With the marketing authorization or new drug application, this is just a blip. You know what blip means? It's a kind of what you call that? A photographic kind of picture on the continuing safety vigilance of pharmacovigilance and monitoring. So what I want to say is it does not stop by getting the authorization or getting the or submitting it. No, it is not. It is continuing process, and that's why you have to consider the post-approval safety assessment. So again, many times after I submit all the data and after the dialogue with the agency, and I can understand why they have issue with the risk management that we submitted or we committed to, because this is your job. The job of the agency is not to get the approval or not to give the approval. The job of the agency is the patient safety. They look after the public health care. Therefore, a sponsor need to realize the perception of the agency and where are you coming from? What is your focus? And so on and so forth. And therefore, agency, many times they give you what is called, you know better than me, the conditional approval. So I'll give you the approval with one condition, that you will do this number of studies I need this number, this kind of data, and what is the duration, what is the population, and what is the dose, and so on and so forth. And here is a question for you guys, so be ready. So in the medicine development, post-approval, what contributes to the safety assessment of a product? So just to give me one thing. What contribute to the safety assessment of a product post-approval? Yeah.
There are no comments yet on the chat.
Okay. I
Giving
will
them
not.
maybe a
I
few
will not go
seconds.
forward. Yeah. I will not go forward. I will wait.
Come on, students, you can type in your adverse events.
Okay. Very good.
ICSRS,
Okay. Individual case.
PSURs.
Yes.
Yes.
There's something I'm waiting for.
PSURs. BRA. Missing information from the study.
That's good. Oh, guys, you're very good students. You're very good. You know everything. You don't need me to tell you. Excellent. So all of these are the contribute to the post approval safety. All the activities in the post approval stage, everything, whether it is what you call it, the SS. I forgot now. The long-term safety clinical trials, real world data, all of that, all this post-approval stage, and all the stuff that you said, adverse events, reported adverse events or serious adverse events, spontaneous, especially the spontaneous one. And this is very important. So why the post-marketing adverse events collection is important? During the drug development process, which goes around 15 years, you are developing this drug, and you haven't got enough of adverse event reporting? Can someone answer this question? No?
I'm waiting for the chats. There's nothing yet on the chats. Okay.
Right.
I
Yeah.
have got a response saying larger population.
Very good. Yeah.
The drug is now used broadly.
Yeah.
Some didn't really get your question. Maybe if you can repeat it again.
Okay. So here we said that it is used broadly, larger population. This is what I was after. In the drug development process, 15 years, say 15, 17 years, how many patients exposed to your drug? Give me a rough number. How many patients? 100,000? Higher or lower?
In a clinical trial, I believe they will all say it's a smaller population.
Yeah, smaller. I just want to put it into
Okay.
perspective. So if we say
There's a
100,000, is it higher or lower?
Okay, we're having comments. Smaller for phase 4 studies, lower than 100,000.
Lower. So lower like 50,000, higher or lower?
Lower. That's the response in the chat.
25,000, higher or lower?
Lower. That's the
Lower.
response on the chat, yes.
Okay, 10,000, higher or lower?
There's a 10,000 and there's a lower.
So it is actually, it could be lower than 10,000. And as you know, guys, because you are a very good students, 10,000, you cannot really get a grasp of any signal. If you get a signal, it means this drug is very bad. It shouldn't be even go for approval. But in order to get a proper signal, you need a larger population, so 100 and above, and you cannot get that until the drug is in the market. Okay, that's why the post-approval stage is very, very important. And I would say that the safety assessment or the benefit risk assessment is even more important when the drug in the market than before the market, when it is still in the development. So we talked here about the PSURs and the breeders. And so what are the objectives? So the objectives here is that you always, as we said, that these are accumulative. Accumulative means what? That the knowledge that you know yesterday is yesterday. Today you know more. So that's why it is accumulative. And when you have more information, so now you have to change your information. This is not written in stones, guys. It is not written in stones. And all these documents are living documents. So they keep changing and keep updating. You add to it or you take out of it. So it is not just that you add information, but sometimes the information you added before and the assessment is no longer valid. So you take it out, okay? And so on and so forth. And you always evaluate the alignment with the previous knowledge, and this is what we said, and the new data you added to that. And then the analysis as well, critical analysis of the risk benefit assessment. And with adding this analysis, sometimes regulators say, well, I am not 100% happy with your analysis, okay? The analysis could be skewed this way or the other way. So this means, can I ask for more analysis? Yes, you can. Can I ask for different analysis? Yes, you can. Can I ask that you give me all the data and I do my analysis? Yes, you can. All right? So you need to think about that. When it comes to this, you are a scientist and you need to make your opinion on a scientific basis, not just how many documents has been submitted and is it with check boxes, is it done or not, is it full or not? No, you need to do your own analysis. Let us look at something here. Peter talked about the risk management and mitigation. Risk management and mitigation. What is this mitigation aspect? And why it is not elimination? Why it is not risk stopping? Why it is not risk abolishing? Can anyone answer that? Why? Why it is mitigation, not elimination?
Sorry, Asim. Maybe we might have to take the questions at the end of the session.
Okay.
We are running a bit over time, and I see you still have quite a few slides to go through. If maybe you can, yeah, we will see how we go about it. Thank you.
So
sorry about that.
No. You cannot eliminate risk. So you cannot ever eliminate risk, but you can reduce the risk. Okay, there is nothing without a risk. So in the risk management aspects, the background here is we talked about that small number in the clinical trial, restricted population, comorbidity, because all of these are governed by the inclusion-exclusion criteria in the protocol, where the sponsor was in control of what to get into the study and what not. Therefore, this is at the time of the authorization. When medicine product is authorized, the benefit risk is judged positively, of course, for that target population. Otherwise, you're not going to get your market. But for that, you need to strengthen your pharmacovigilance activity. Of course, you need to do that. And post-authorization, you need to carry on after that. And these issues are addressed in the form of the plan. So you say that this is the benefit, okay, everybody is happy with that. And this is the risk, and this is how I'm going to manage or mitigate that risk. The risk identification and characterization is very, very important. And agencies usually ask you to present that and go back and repeat it again and repeat your risk characterization and come back with a plan, because this is not complete and this is your right to critique review this risk management plan and mitigation. The risk management, 3 stages for the risk management. Number 1 is the characterization. Of course, you need to characterize that first, and then you plan your pharmacovigilance activities according to the characterization, according to the profile that you have in the first stage. And this is how you increase the knowledge in general about the safety profile. And then after that, the planning and the implementation of this risk minimization and mitigation. Number 1 is to characterization of the safety profile. Number 2, that you plan your pharmacovigilance activity, and then you implement this stuff for the risk minimization. Okay. Now, the objective of this plan is what? Number 1, identify the safety profile, indicate how to characterize further the safety profile. So it is not, as I said earlier, is not a done deal. So how you forward to characterize further the safety profile. And also you need to document the measures to prevent or to minimize. You need to document that. What are you going to do? You need to document that. Post-approval obligation or post-approval conditions need to be imposed as a condition of the marketing authorization. And this, there is no negotiation on that. Patient safety and the public safety is paramount. It has always to come first. What is the structure of this plan? It's a long plan. You know this better than me with all these sections, but I'm going to talk about 2 sections. Number 1 is the safety spec and the risk minimization. For the safety spec, and you will have all these slides available to you afterwards, so you could go through it. And I'm happy to answer any questions afterwards. You can just send me email if there is something not clear enough. So here about the purpose of the spec, to provide the synopsis safety profile, summary of the important identified risk, important potential risk, not just the risk, potential risk, and also the missing information. And it should also address the population potentially at risk. So it could be that some subpopulation potentially at risk, not all the population. You need to identify that, and you need to talk about your mitigation in that section. Right. The safety specs here, I was still talking about safety specs or how you're going to use it and so on. And right, the risk minimization. This is part 5. So this is the end. And how to evaluate the need for risk minimization activities. We said that you cannot abolish risk, that you need to minimize that risk with your, so on the basis of the safety specs that you identify in the process before, now you need to provide an evaluation, the need for the minimization. And not only that you provide these measures or the activity, but you need to satisfy the regulators that these activities are actually effective. Okay, so it is not enough that you say, I'm going to do X, Y, Z. Tell me that this X is going to minimize how much and how, and you tested it, and it is good enough, and so on and so forth. All right. For some routine risk minimization is not enough. So it is also, if you are not satisfied as a regulator, as an assessor, you ask for additional risk minimization activity. And this is and this is normal. It is not, so don't think that you are asking something difficult. Again, here we're talking about the routine risk minimization activities apply to every medicinal product. So you have here the routines one, the SMPC, the labeling, the leaflet, the legal, and so on. And sometimes the package size, of course, in cases of addictions, potential, and so on. So you need to have the pack is a smaller a smaller pack, and so on. The design and the package and the information itself, again, how to prevent or how to minimize the medicinal error. Additional risk minimization activities. So this is above and beyond the routine ones. And remember what I said earlier, that not only that you have the activities, but also you need to assure me that these activities are effective enough. Let me here summarize, and I apologize for going over time. The risk orientation can influence the benefit risk assessment, of course, and can change over time and change from country to another and change in the same country from one population to another. And we said that. Contributor to the safety assessment of a product. Post-approval, you have the activity in the post-approval stage to ensure the continuing monitoring, periodic safety update report, and the risk management and mitigation. With the periodic safety update, you say, me as an assessor, can I ask for shorter period to provide this report? Yes, I've seen it before. Some assessors said, okay, I'm not going to wait for 6 months. I need something before that. Yes, fine. You know, it's not. Okay, benefit risk assessment ongoing. As we said, it is not written in stone. It's a living document, and you have to add in or take out. At the time of the authorization, information on safety myths is limited, and we know that because of the smaller sample size that this information is coming from the clinical trial. That's why, and also the restricted population because of the protocol and so on. That's why you need a robust pharmacovigilance activities, which is essential to identify new and characterize new information and to mitigate this risk and potential risk that you haven't identified before. I think this is my last slide, and I will stop here.
Thank you. Thank you very much, Assam,
And I
for that
apologize.
presentation. Not a problem. You are forgiven. It was an interesting topic. We will now go to the bio break quickly, and when we return at exactly 15 minutes past the hour, we will go into our breakout rooms. I know it's very tight, but a quick break, and then we go into the breakout rooms. Thank you.
Thank you very much. Thank you.
Welcome back, everyone. We will now go to our breakout rooms. Everyone had the opportunity to go through the questions on the Moodle platform. We've seen some of your chats. You will have 10 minutes in the breakout rooms. So we basically just debriefing and discussing, and then we will return and we'll have a moderated discussion session led by Peter. If we can put the rooms up, Steph. Welcome back, everyone, from that speed session. I will now hand over to Peter to lead the discussions and maybe summarize for us. We only have 10 minutes for this session. Thank you, Peter. Peter? Is Peter with us now?
Hi, Anat. They're still coming out of the rooms, it seems. They're coming in.
Oh, okay. Welcome, everyone. Is everyone back now? Peter,
Yeah.
I had already started talking and I realized you were still finalizing with your team. I will now hand over to you to lead the session and maybe just summarize for us as well. We are short of time, so we have about 10 minutes to conclude this session and move on to our next speaker.
That's okay. That's okay. We've agreed that we would have a fairly short plenary session this time, and indeed we will have a plenary session. And we're not at this time going to ask each rapporteur to feed back in turn. What we're going to do is to ask the questions, and then it will be very good if those people who have prepared some answers, and I have seen some on the emails as well. It would be very good if you could make some of those points, which will actually help to summarize the situation, really. So can I straight away go straight in? Oh, we've got a video here. That's well, okay. Right. Can we go straight away and ask the first question? What is your agency's benefit risk assessment for this product, considering the nature of the disease indication, which was psoriasis, the potential benefits for the patients, and the possible side effect profile? So without more ado, would somebody like to make a statement about that to lead the discussion in with everybody? And let's do it. And
okay, so this is Jessica from
yes.
Group Room 3.
Thank you very much, Jessica.
Looking at the question, I think we are going to have a positive benefit risk assessment. The nature of the disease is an autoimmune disease which has no cure, which has nothing to, cure it. So we are looking at measures that can reduce symptoms and improve overall quality of life. And when you look at the drug in question, it provides a lot of relief, and the relief it provides is long-standing. And that is going to be very beneficial to the patients. And as much as it will be a positive benefits risk assessment, we'll still be looking out for some of the adverse events that the patients will experience, that overall group thinks that is going to be a positive benefits risk assessment. Thank you.
Jessica, thank you very much for that wonderful contribution. That really summarizes quite a lot of things. Because you've drawn out straight away that it's not just the drug that you've got to look at. You've got to look at the patient, and you've got to look at the condition, and so forth. So benefit risk assessment is not a straightforward addition mathematic. It's basically looking at the context of what you're assessing as well. So would somebody else like to also make a comment on question 1, or shall we move to, well, question 2 and question 3 actually were there just in case we couldn't find an answer to question 1, but so far you're doing very well. Could I ask somebody else to make a statement about, there's lots of agencies represented here. What is your agency's benefit risk assessment for this product, considering the nature of the disease indication, the potential benefits for the patients, and the side effect profile? So somebody else.
Okay, I think Jessica has already said a lot. So
yeah, okay, thank you very much, Zillian.
Okay, yeah, so we had said that we look at the disease, like she said, the disease, the treatment. Does the treatment meet the unmet clinical need? We look at possible past medications that the patient has been on, and have they to prove that they have not met the clinical need. So yeah, I think that's all I can add because she's said everything so far.
Okay, that's fine. You added actually something there, which was the unmet clinical need, yeah, which was very useful. Yes, thank you. Somebody else anybody else? Do we have?
Well, from group 5, which was my group, we also came to the same conclusion
Right.
as the others in terms of a positive benefit risk assessment. But in addition, in terms of the side effects, the group identified that although there were some risks in terms of the side effects, they could be easily managed and there would be an improvement to the quality of life of the patient. Thank you.
Right, and the quality of life, yes. Okay, thank you. Thank you very much. That was Tombi.
Yes, that was the.
Yes, thank you very much, Tombi. Fenyo was going to speak, I think.
Yeah, I think. Good afternoon.
You've looked at this and wrote an answer. So tell us.
Yeah, so we also reached the same conclusion as the other groups for a positive benefit risk because the disease is not pervasive and there are not so many therapeutic options available. We also looked at the fact that the disease is affected by ethnic variability and therefore we may, to some extent, need targeted medications, and the side effects identified can also be managed, and we believe this medication would improve the quality of life of the patient, so we would give it a positive benefit risk. So
Thank
that
you.
was for question 1. Yeah.
Right, thank you, Fenyo. You made the point as well that there was a need for creating as much diversity of treatment in this condition because of the variability, the ethnic variability of it, and that's a good point too. Right, okay, and Natombi, you talked about quality of life. That's an important thing to say as well. And yes. Anybody else?
Have we had anyone so far for question 2, so that we try and move with the session? Maybe we can now deal with the other questions, Peter.
Yes, okay, we can do, yes. Basically, this was bringing in a bit more granularity into the terms of what data we would need to collect to actually advise on to actually drive the benefit risk assessment. I mean, what you've actually done so far is taken the case study that was presented to you and looked at the various issues to do with psoriasis in your community and the treatments that are available and looked at the benefits and risks from that point of view. And I think that's a very useful way of doing it without actually going into too many assessments and mathematics. But the next questions actually relate to what sort of data, what data are you looking for in order to reach a benefit risk assessment? And it may take longer than we've got to answer that question actually in Unati.
Most probably. Maybe we can rather summarize and just point out the things that we were expecting, because the students have already tried to answer the question on the Moodle platform as well.
Yes, indeed. Well, I mean, to summarize this, basically, the way you've done it, the important point is you've considered the patient, the disease, the treatments, the population and the ethnicity issues, and you haven't just focused on a list of harms and benefits, which is another way of doing it, of course. And if we were to use the template approach to actually do a very exhaustive benefit risk assessment of this product, possibly for regulatory reasons, for regulation, then I think we would be collecting much more systematic points about harms and risks, which may not yet have been expressed in the adverse events and adverse reaction list, but maybe be part of the list that Assem El-Baghdadi showed on the possible harms that come because of medicines. So in doing a full benefit risk assessment, I think there would be many, many more variables and many more situations that we'd have to consider in making that assessment. But the ones you've made so far have raised the important point of considering, not just the drug and its side effects and its benefits, but also the patient, the disease, the treatments available, things like quality of life and ethnic variability and the psychological effects and so forth, and the fact that this benefit risk is a very changeable. It doesn't have to be a consistent point either. I mean, benefit risk assessment could be changeable depending on many different things. If this particular product was the only product available, that may change the benefit risk. If the product works and even if it has some side effects, it may be the only treatment available, in which case the benefit risk assessment would change because of that. So there's a huge number of factors to consider here, far too many for us to discuss and analyze at the moment. But the takeaway message from this is that you do have to look wider than the actual drug profile itself to the efficacy and safety profile of the drug itself, to come to a proper assessment of benefit and risk, which will lead you to a more valid and useful regulatory assessment and decision. So I think I'd just like to leave it there, really.
Thank you very much, Peter, for that summary. And we are now going to hand over to our last speaker. Thank you very much, Peter. Anyone who has any additions regarding the breakout room questions, they can add on the chat and we will look at them, and then Peter will help in answering, and some of the other faculty members. Now I would like to welcome Doug, our last speaker for today, who will present the hot topic, one of the hot topic that has come up in most of the lessons that we've had so far, and that is real world evidence. So Mark is the Deputy Director of Integrated Development at the Gates Foundation. He provides guidance on product development, drug safety, drug modeling, and simulation. His expertise extend to biomarker discovery using real-world data and machine learning. And just to remind you, if you want to view the full bios for our speakers, you are welcome to go and view the Moodle platform. Thank you very much. Over to you, Doug.
Thank you very much, Renati, and welcome everyone. I'm grateful for the chance to talk today. At Gates Foundation and in my previous employments, I've been engaged in real-world data analysis and machine learning using confidentiality-protected electronic health record-derived data in support of not only medical products development and regulatory submissions, but also pharmacovigilance. This is essentially a follow-on to the remarks that Peter and Assem had earlier. And in anything that involves uncontrolled observational data, like real-world data, it's essential to mitigate biases and confounding. And that's a good bit of what we'll cover in the next few slides. Let's see, there we go. Well, there are many ways that real-world data can provide evidence to support and accelerate decision-making in regulatory practice, especially post-market safety surveillance. In recent years, and especially during the pandemic, there's been much activity using real-world data for identifying opportunities for label expansion and real-world data arising from de facto off-label use of drugs or concomitant on-label usage in patients who have more comorbid conditions besides what the drug is being used for, such that an unexpected benefit may arise, and sometimes some unexpected safety issues can also arise, part of the ongoing benefit risk assessment that we've been discussing. Especially too, real-world data can help in quantitative design, not only of informative clinical trials, but also in a variety of pharmacovigilance signal detection use cases. Ideally, real-world data provides reliable evidence of effects in the large as-treated populations and the post-market registration context, with cohorts in the hundreds of thousands to millions, usually, far greater sample size than is practical in conventional pivotal trials. And real-world data, therefore, can have power enough to answer questions about safety and about effectiveness, dose range adjustment, regimen optimization if there are 2 or more drugs that are used concomitantly, and also about cost effectiveness and quality of life. These sorts of things are important in all of your countries in the years ahead, just as they are important elsewhere. Several years ago, the US FDA released a guidance on real-world data. The link to this is in the references at the end of the slide deck on the Moodle platform. The guidance is notable for including a number of specifications to assure the quality and reliability of real-world data that may be provided by sponsors to regulators like you in support of various pre-authorization submissions or in connection with post-authorization pharmacovigilance or safety surveillance. The context of observational research using large real-world data repositories makes possible a new kind of mindset, and that is part of this future vision, I would say, a hypothesis generating one rather than just inferencing and hypothesis testing. Discovering patterns that nobody's ever thought to study before. It's important to recognize, though, that despite many tens or hundreds of thousands of findings and observations, various types of laboratory tests and formulary items there are in the GDPR compliant electronic health record derived real world data repositories. These repositories do only contain the information about things that clinicians thought about, that they documented or actually did. If something's very uncommon in clinicians' thinking and actions, then there might not be much data about that to analyze in the RWD. Basically, though, we can perform in silico, in the computer, with real-world data, most of the analyses that we're accustomed to doing in randomized clinical trials. We can ascertain endpoints, efficacy ones, as well as adverse event or safety endpoints in the observations and lab values, and we can do signal detection. A large number of things that are measured in RWD, many of them measured repeatedly and longitudinally in tens or hundreds of thousands of patients, represents a distinct advantage over individual case reports. If the data available are large enough, you can do statistical analyses that discover and measure the drivers of effectiveness and safety and causal relationships. A large cohort size in real-world data affords the luxury of exploratory and discovery-oriented analyses and Bayesian prior statistics to support simulations. The high dimensionality in real-world data these days affords the opportunity to explore possible multivariable relationships that you just didn't have enough data in pivotal clinical trials data sets to do. And it's possible to discover heterogeneity in the treatment effect, or HTE, as FDA and other regulators prefer to call it. And once that heterogeneity is discovered, you can identify clusters or subgroups that merit additional sub-analyses and so on. So in general, RWD is most valuable in estimating the effect size for these kinds of events or situations, and powering simulations to optimize the design of prospective studies of them. For your roles in regulatory agencies, we should talk a bit about the nomenclatures for coding safety. The Council for International Organizations of Medical Sciences, CIOMS, has their medical dictionary for regulatory activities, acronym MEDRA. It was developed in the 1990s and was based on an earlier version created by the UK regulatory agency, MHRA. The International Conference on Harmonization took it on, and an international group developed it. Organization was created, the MEDRA Maintenance and Support Services Organization, or MSSO, to administer and maintain it. There are links to that in the slide deck on the Moodle. The MEDRA nomenclature has 5 levels, more than 90,000 terms. These code sets are part of what you can have in some of the longitudinal real-world data repositories available for you and your agencies to use. So just to give you an idea, here's a list of those top-level concepts in MEDRA. It does include social circumstances, some quality of life terms as well, things related to deprivation or homelessness and the like. But most of these are pretty physicianly or PharmD-ish clinical pharmacologist oriented. The 8-digit MEDRA code that identifies each adverse event type is assigned sequentially. They're for the past 20 or so years referenced in various electronic submissions, and they're aligned with the ICH E2B guideline. But there are complexities here that you'll encounter and are able these days to use in the real world data sets. The different dimensions or axes of the MEDRA and SNOMED CT and other nomenclatures, a multi-axial data model enables each detailed concept to be composed by joining 2 or more other codes. The scheme enables a flexible searching and retrieval from real-world data systems. So here's a simple example just to show a lower-level preferred term for influenza, how it is a descendant of 2 axes, one for respiratory and chest conditions, and one for infectious disease. Here's another simple example which shows a low-level term for hyperventilation, how that might arise for either of 2 reasons or causes, one involving psychiatric distress and the other involving cardiorespiratory physiology. The complexity of the multi-axial mapping is in the 5-level hierarchy, more complex you might expect. This example for diabetic retinopathy as a adverse event or outcome is shown. The relationships of the outcome to the disease process and its sequelae and its dietary and pharmacological treatments are possible to explore quantitatively in the real-world data when one has a code set and a detailed model representing things like this. Despite the many advantages, nothing, not even the MEDRA nomenclature, is close to perfect. So when you're using real-world data systems or evaluating or specifying, procuring one for your agency, it's wise to take into account the need for mapping between nomenclatures so that the things that you can do are not confined to any one supplier, any one data system, and the engineer's design ideas at the time it was introduced. Note that there are aspects that are not supported by MEDRA code set. Probably the biggest limitations, in fact, are that MEDRA codes do not account for the frequency, the duration, or the severity of the adverse events, only the presence or absence of them. So, as with other things for severe or other adverse events in real-world data, it's important to remember that severity is not the same as a serious event, which has more to do with the duration or irreversible nature of an AE. You can have a severe AE that is reversible, and you can have a serious AE that is only moderate in severity. So where are we these days? For one thing, we want to detect drug-related safety issues with real-world data. Traditionally, that's been done passively with statistically measuring the disproportionality of reporting rates from individual case reports, spontaneous adverse drug reaction reports. But it can be done actively in an ongoing longitudinal way with real-world data that's updated weekly, monthly, or quarterly. Chi-square and odds ratios are the classical statistical ways this is done, but there are other methods that have been introduced over the past 20 to 30 years that are supported by free and open source software packages that you can use, the R system, for example, as well as commercial packages that sponsors and you and your agencies use. Oh, there are a few slides, some with some equations in them for your interest. I think for the reasons of time, we'll omit these, but basically you're accumulating accounts for various events in the rows and for the relevant drugs that might be used and be associated with adverse events, and then the estimation of the rates and whether they're disproportional or not is part of what the algorithms produce. Here's an example that I did myself some months ago in the context of hospitalized patients with COVID to see whether there were any adverse event signals connected with ivermectin. And naturally, for moderate to severe COVID patients admitted to hospital, there are a wide range of other medications that are used in that context. I surveilled 40 of the most common ones and looked for 177 different adverse event codes from the real-world data, lab tests, and other information, not just ICD-10 codes or MEDRA codes. And here in just a few dozen lines of SQL code, you retrieve the data from the real-world repository that you need. I mean, this is not rocket science. It requires careful work, but it's not terribly complex to do. In your regulatory role in your country, it's important for you to have a firsthand sense of how you or your statistician colleagues can go about work like this. So you retrieve the data that your SQL queries generate and get your cross tabulation with accounts N for each of the drug adverse event pairs. This table can be huge, hundreds of thousands of rows typically. And then you run your pharmacovigilance software. The code that's shown on the screen is a simple example of that. And what you come out with is an indication that for the ivermectin, in this context, you do have the presence of a signal, a proportional reporting ratio of 24, very high, for hepatotoxicity, alanine aminotransferase, greater than 5 times the upper limit of normal. So there are a variety of causes for this, perhaps related to liver metabolism, CYP isoform, liver enzymes, and so on. But it doesn't take a tremendous amount of work to be able to do signal detection and validation with these things. The proportional reporting rate is the oldest of the signal detection metrics. There are other things that in the supplementary slides in the Moodle version of this PowerPoint you can take a look at for those of you who are interested. And these bullets basically illustrate some of the questions that are raised with this sort of a pharmacovigilance example. There are other things here too, but one usually uses a mixture, 2 or more of these several signal detection algorithms, empirical Bayes, O5, multi-item, gamma Poisson shrinker, Bayesian confidence propagational neural network. All of these acronyms look pretty unusual, but relate only to a few lines of code that you or your statisticians can run and do repeatedly. So you're monitoring, as Peter and Assem said, the evolving signals as the context changes or the population in whom a given drug of interest is used to change. So I think for reasons of time, I will stop sharing there. Very grateful for the chance to talk with you today and look forward to whatever questions, both while we're active today or via email later. Thank you very much.
Thank you very much, Doug. This has been indeed a hot topic in the other previous sessions, and we are really happy to have had you give us the talk. And I would like to encourage the learners, the students to actually go and read up on the slides that Peter, Assam, and Doug have prepared, and just to enhance the knowledge, and you will find value and will benefit from the material they have prepared for us. We really apologize for the time, and we have been pressing all our speakers, and I'm sure they would have all loved to share just a bit more with us. Before I close and take and remind everyone about the other lessons that we are still going to have, I would like to give an opportunity to Peter, Doug, and Assam, just to have your final words in closing. Over to you, Peter.
Thank you, Unati. I hope that this session with all three presentations has brought together thoughts and ideas about drug safety and its implications for not only for producing clinical data of importance for regulation, but also for obviously a continuing effort to keep patients safe and to monitor the effects of our medicines. Because I can't emphasize more the fact that this is a continuous process which never stops, and therefore all the measures we make are also very variable. So the things we've talked about today are changing and are variable and can be and must be repeated at regular intervals to make sure we're up to date with the latest in the product profile. So, with that, thank you very much to all to our colleagues, Doug and Assam, for their contributions, and to you, everybody in the audience, for making such good and valuable comments when requested, and also to you, Unati, for chairing so wonderfully this meeting, and of course others behind the scenes as well, helping to make this all work. Stephanie, so thank you.
Peter. Thank you, Peter. A word from you, Doug. I see your camera is on, and following you, it will be Assam. Thank you.
Yeah, thank you, all of you, for your excellent contributions. The diversity of views here and the ways in which safety depends on the context, as Peter and others of you have said, is so important. Again, thank you for this time together.
Thank you very much. Assam, I will now hand over to you, your final words. Thank you.
Thank you very much for the invitation, and thank you for the organizer. And I'm sure a lot of work is in the background that we don't know about. And so thank you very much. The students, you are great guys. You are full of knowledge and experience. And I can see your comments keep coming, and I promise I will answer as much as I can together with Peter. And if there is something, the message home is, guys, you do not have to accept assessment of other agencies as a gospel. You have to go through the assessment yourself and ensure the safety for your patients or for your population. So if another country accepts something, you absolutely right not to accept it. You are the best to know your population and the background of the country that you are working on. You are scientists, you are not a clerk. You do not check boxes to make sure that the dossier is complete. You need to make your own assessment as a scientist, and everything needs to be justified scientifically. Do not be apprehensive from going back to the sponsor once and twice and ask for the data and ask for more data and so on and so forth. So with that, thank you again for all your efforts. I really enjoyed today. Thank you.
Thank you very much. We really appreciate all of you, all our speakers, and thank you to everyone who has participated today, to our students. And again, I would encourage you to go into the Moodle platform. If you do have any other questions that you were not able to get them answered in this session, you are more than welcome to add them in the forum, and we will forward them to the relevant people if we are not able to address them. So we are now at the end of the session. We are almost done with the course. I cannot believe how time has flown. So next week we will have same time. We will have lesson 12, and it will be led by Leander Fontaine and Regine Lenhart. It will be on marketing authorization and product information. We're looking forward to having you join us again. A big thank you to our presenters. Thank you. We really appreciate your time and all your contributions. Students, don't forget to go to the Moodle platform for all the additional slides that our presenters were not able to cover today. Thank you, everyone. We will now close the session.
Thank you. Bye.
Thank you. Bye-bye.
Thank you. Bye.